Alright everybody, welcome back to Lab 5 of PLPA 303. Today we're going to be working with Agrobacterium, and we're also going to get a little bit of an introduction into mycology. But before we get into the lecture, I just want to give you some general announcements. Again, continue to read the questions carefully on the assignments that you guys are turning in. Make sure you answer all the questions fully, especially when you come to those questions that have many points, because if you only answer half the question, I can only give you half the credit. And if it's like a 15-point question, then you already lost almost a letter grade there. So please read the whole question and answer it all. And I've been also getting some emails about, well, there's been some confusion on when to turn in some assignments, especially those assignments that are due 24 hours afterwards. Were you guys all able to submit on time? Okay, so you should have multiple submissions on eCampus, and you only need to submit the portion of the assignment that is due. So I know for some of the assignments you have multiple parts due at different dates. It's okay if you don't submit the whole thing completed, because obviously you won't be able to complete the whole thing. So long as the portion that is due is completed, we'll give you full credit for that. And then you can resubmit it. You can resubmit the second half or whatever portion is due later, at a later time when it's due. And if you still don't understand, in the very beginning of your lab manual, you should have this very kind of confusing, I'll admit, assignment turn-in guide. As you can see, the labs don't coincide with the assignment numbers. We've gone over that. But also keep in mind, if you don't understand what's due or when it's due, you can see here, for today's lab, we're going to have 3 parts, all from different assignments, and they'll be due on different dates as well. The reason everything's all screwed up is because of COVID, so we apologize for all the confusion. Just hang on to this and just follow it closely. And even then, at the top of the assignments, for example, a few weeks ago, we had assignment number 3. It'll tell you right then and there when something is assigned and when it's due. So if there's any confusion, just go ahead and look at that, or just email me or whichever TA is in charge of your section. Alright, let's get into the actual lecture then. So before we start talking about Agrobacterium, we first need to understand what a plasmid is. Do you guys know what a plasmid is? Who can tell me what a plasmid is? Anyone? Slow Monday. A plasmid is just circular DNA. Here we define it as being self-replicating, meaning it has its own origin point, which is where a DNA polymerase is going to come in and start to replicate it. It's also extrachromosomal, so here we define it as not being part of the bacteria's genome. So it's non-essential. The bacteria does not absolutely need the plasmid to survive. A bacteria can also have single or multiple plasmids within their cell. So each plasmid can encode something different, or they can just all be copies of the same thing. It's circular DNA or genetic material. We already know that. It also has to be heritable, meaning that it can be passed on to the next generation of bacteria through fission. And it also has to be, we define it here as also being transferable, meaning it can be passed from one bacteria to another through conjugation. And here in these electron micrographs, you can see the linear DNA just looks like a regular strand of hair, even though here with the electron micrograph, we're looking at this image blown up several, tens or hundreds of thousands of times. And you can see the plasmid here looks like an open circular DNA. By open, it doesn't mean that it's linear. It just means that it's open up kind of like a rubber band or like a hula hoop. And then you've also got supercoiled DNA, which is also a plasmid. But if you ever taken a rubber band and you started to twist it or scrunch it up, you'll see that it kind of balls in on itself. Well, DNA does something similar in which it becomes all coiled. We call that supercoiled DNA. And then just more images of the plasmid down here. Alright, so Agrobacterium. We learned about Agrobacterium before we worked with it a few weeks ago. Who can tell me what does Agrobacterium cause in the plants that it infects? It causes crown gall, yeah. It causes sort of a little tumor-like appearance on the stem of the plant. It only infects dicots mainly, and it only infects wounded sites. So bacteria are not like other plant pathogens in which they force their way into a plant. They'll get into plants either through natural openings like stomates or lenticels, or if the plant is wounded, then the cells inside the plant are completely exposed. So bacteria can also start to swarm and colonize the plant itself. And Agrobacterium is naturally found in the soil. That's the reason you see the crown gall near the soil, because it doesn't have to go very far. With Agrobacterium, once it establishes itself, it doesn't really cause a systemic infection. It's kind of localized to that area. Kind of like how last week we talked about those sedentary nematodes. However, with the plant, it also starts to cause more. The bacteria starts to cause more growth of the plant in that local area where it infects. So today we're going to learn a little bit more about what causes that gall, why we get so much growth of the plant. So Agrobacterium is also known as nature's plant genetic engineer, because it is able to literally modify the genome of its plant host. And it does so by inserting a part of a plasmid that it encodes. So this is not part of the Agrobacterium's chromosomal DNA. This is just something extra. It's got something called a TI plasmid. TI stands for tumor-inducing. And it's also an important genetic engineering tool, and I'll show you how we managed to take advantage of this later. But here's the natural, or simple, I should say, diagram of this TI plasmid. You've got a virulence region, which is what encodes some virulence factors that I'll get into. You've got your left border and your right border, and in between it, you've got your tDNA region. You've also got a couple other things, your origin of replication, which is often abbreviated ORI, O-R-I, and you've also got an opine catabolism site. So we'll talk about what opines are in just a minute. And just so you guys can see what a plasmid map looks like, these you've got this kind of line that goes around the entire plasmid. That's just not junk DNA, but rather it doesn't really encode anything. So it's not terribly important other than for the structural integrity of the plasmid. And then you've got these raised kind of boxes around which you have several genes. And if you guys recall what a whole gene looks like, you'll have a promoter, you'll have the coding sequence. So the promoter is where the RNA polymerase gets attached to the DNA itself. Then you've got the gene, which is what's transcribed and later translated after it's been modified. And then you've got the terminator, which is where the RNA polymerase falls off. And just so you can further see what those plasmids look like, here in working with labs, we often work with plasmids. And you can see that the genes that it has encodes for something. So for example, ampicillin resistance. Ampicillin is an antibiotic that we use for selective media. So if we want to put something, or if we want a protein to be created by a bacteria, we'll first get this plasmid, we'll put the sequence for that protein within the multiple cloning site, and then we'll grow it on media that has ampicillin on it, so that any bacteria that does not have this ampicillin resistance gene is going to die, and you'll only be left with bacteria that produce this protein. And you can see there's different types of origins, of ORIs, origins of replication. They're not all the same. It all depends on their sequence, and their sequence can determine how often these plasmids get get replicated. And these are just restriction enzymes. You guys don't have to know about all this, but this is just to show you that plasmids are very common in molecular biology lab settings. So let's take a look at the infection process and see how Agrobacterium is actually able to infect and create this gall in its host plant. So first the bacterium enters the host plant through a wound, either through natural opening or through a wound. And this wound can be created in various different ways. You know, just gardening, maybe an animal comes and hits it, or an insect feeds on it and somehow the bacteria gets in. What the bacteria is able to do is inject the tDNA region of the TI plasmid into the plant. So what the bacteria will do when they sense that a plant is wounded, when it releases this enzyme, or excuse me, this molecule called acetosyringone. Acetosyringone is normally not found in the environment, but it's released when a plant is wounded. And so Agrobacterium in the soil will swim towards that wound and get into the plant in that manner. It also activates parts of the virulence region. The virulence region is what's going to allow the bacteria to actually create sort of like a needle, which is called a type 4 secretion system in this case. And what the bacteria does is it gets right up to the plant cell, and it stabs the plant cell with this needle, this type 4 secretion system. And the tDNA region of the TI plasmid is replicated, just this part, and it's also and it's passed through the secretion system. And so it's kind of like a needle, like what we'll be doing today. Once that DNA is inside the host plant cell, it finds its way into its nucleus with some proteins that are also encoded by the Agrobacterium. And it'll actually insert itself into the plant's genome so that this tDNA part will actually become a part of the host plant. And so that's why we call it a genetic engineer. We're literally changing the DNA of the plant, or the bacteria does that. Now, the reason the Agrobacterium wants to introduce the tDNA region into the plant is because it has several proteins that will produce auxin and cytokinin. Do you guys know what auxin and cytokinin are? Have you heard of them before? They're plant hormones that are responsible for causing plant growth. So now the plant will start to produce more of these auxins and cytokinins within that area, within those cells that have been transformed by the tDNA. And so the plant will start to invest more and more sugars and energy into those cells because it's artificially creating all these plant hormones that tell it to grow. What's important is that you also have these opine, opine creating enzymes. Opines are similar to amino acids. They're not amino acids, but sugar molecules, and they're molecules that the plant cannot use. So it's a source of carbon for the bacteria. However, only the bacteria can use it and not the plant. So it's kind of devious in how this bacteria is able to take advantage of its host. Now, some scientists back in the late 1970s were able to figure all this out, and they said, hey, what if we were to re-engineer this plasmid and take out this, for example, the auxin and cytokinin and opine producing parts, and instead replace this with a gene that we're interested in, such as a gene that will give plants resistance to, I don't know, fungus or an insect. And that's just what they did. They were able to use, to create a construct being some type of TI plasmid that contained some gene which made the plant resistant to infection by other pathogens. And they transformed the plants using Agrobacterium. So they put this TI plasmid into Agrobacterium that did not have a TI plasmid, and then they transformed plants with that bacteria containing the gene of interest, and they were able to get some, plants with improved tolerances to pests, pathogens, chemicals, and even abiotic stress. So this is a method by which we're able to create GMOs, genetically modified organisms, or in this case, genetically engineered crops. Some of the disadvantages of doing this, though, of creating GMOs, is that the gene that we introduce into the plants can sometimes get into other plants that we're not interested in transforming. So, for example, if you guys have heard of Monsanto Roundup, back a while ago, Monsanto introduced this line of Roundup-ready crops. Roundup is an herbicide, meaning anything that is not resistant to this chemical, this herbicide, will die. And it was used pretty effectively to treat weeds growing around crops, because you could spray this herbicide on everything, and only whatever was resistant would survive. However, these Roundup-ready crops, I don't know if they were made specifically with Agrobacterium, but part of one of the negative side effects of creating this GMO is that some plants, some wild relatives of the crops that were Roundup ready, that were resistant to that herbicide, they also became resistant to herbicide, because they were able to cross some of that pollen from the genetically modified crops got into the carpels of those wild crops, those kind of herbs, not herbs, the weeds. And so what you ended up with was a genetically engineered weeds and other things like that. So there are some issues that can arise when producing GMOs, especially with crops. So that's just something for you to keep in mind. And just to show you guys what I do in my lab, or part of what I do, is we took a TI plasmid and we replaced it with the coding sequence of a virus, rose rosette virus specifically, so that when this is transcribed, when you get the RNA portion of the sequence, you get viral proteins after they're translated. And we also put in GFP, green fluorescence protein, and we infiltrated the leaves of this tobacco plant. Now this is a tobacco plant under UV light. Normally a plant looks red or kind of dark blue, kind of purplish under UV light because of all the chlorophyll that it has in it. But here you can see it's a bright green, and that's because it's got all that GFP in it. And if you look at the epidermal cells underneath the confocal microscope, you can see that the cell wall and even the nuclei are fluorescent because they have all that GFP in there, because their cells were transformed by Agrobacterium that contain GFP, or that code for GFP. And so we were pretty happy with that. We were able to publish based off of this. All right, so switching gears a bit, we're going to look at what causes disease, or what stimulates disease. You guys have already heard of the disease triangle, and for the host, each part of the triangle has some factors that are important or necessary in order for there to be infection. So, for example, the host must be susceptible to the pathogen. And you also need to have inoculum in sufficient quantities. So with some pathogens, such as some types of bacteria, or rather some types of fungi, the host plant is able to take care of a couple of spores. Not just one single spore on a plant will not always produce infection. But if you start putting millions and millions of spores on the plant, then the plant cannot deal with all the spores at the same time, and so you end up overcoming the plant's defenses, and that's part of how you get infection. Let's see what else. You also the site of infection is important as well. We call this the infection port. So remember, last week we were working with nematodes. Nematodes don't really chew up the leaves of a plant, and even if a plant has its leaves on the ground, that's because nematodes are specifically attracted to the roots of plants. So the site which is in contact with whatever could cause disease is important to know, because you can possibly avoid infection if, for example, you don't have your leaves on the ground, so that you reduce the amount of the plant body that is in contact with wherever the pathogen could come in from. Let's see, some plant tissues are more susceptible than others. Just explain that. And some techniques cannot be used to infect certain plants or the plant part. So, you know, I don't know if you could, but if you were to inject those nematode eggs that we were looking at last week into the plant, I don't know if that would cause infection or not. It'd be interesting to try it out. And of course, the environment is important. Usually a humid environment is what will cause disease, just because it's so favorable to pathogens. We need to also go over a couple of inoculation techniques. You guys did the stab and swab method, so that's kind of a mixture of these two. You can also spray as well. That's part of what we did in a paper we published. We sprayed Agrobacterium onto the entire plant, and the Agrobacterium was just able to enter the plant through natural openings in the host and transform it. You can also do mechanical transmission, in which you put something like carborundum, which is this very fine powder, but if you look at it under a microscope, it's got a lot of very rough edges on it. So when you actually rub a plant with that carborundum, you're causing tons of damage at the molecular level that can allow certain pathogens to get in. Or rather, not at the molecular level, but at the microscopic level. And then you can also use amended potting media, in which, say for example, we want to infect a plant with nematodes again. We can just mix the nematode eggs with the soil and then plant something into that soil, and as it grows, it'll naturally become infected because of all the eggs that are in the soil. So that would be more, you know, if we want to inoculate something to study. There's also syringe inoculation, which is what we're going to be doing today. Let's see, they wanted me to be careful around, or be very specific with the instructions that I give you. So let's see, inoculum is injected into the plant with a sterile syringe, which you guys should have at your desk. Do not use your fingers as a backstop for the syringe. You know, obviously you don't want to stab yourself. The main safety part today is not to stab yourself or anyone else with the syringe. I don't know if syringes are considered controlled substances here in Texas, but where I'm from they were, so please don't take any of the syringes home with you, or you will get in trouble. I'll show you guys how to use the syringes safely and where to dispose of them. Each person's also going to get their own plant to inoculate. Alright, so that's it for Agrobacterium. We're going to switch gears a bit and get into mycology for next week's lab. So mycology, as its name suggests, by the Latin name, myco means fungus, ology means study of, so this is a study of fungi. And it's pronounced fungi, not fungi. The British pronounce it fungi, which is fine as well. Just don't call it fungi or fungi. It's fungi. And you can see all the fungus-related stuff that we're going to be working with usually has myco somewhere in it. Let's see, reasons to love fungi. I'm sure you guys already know. The reason we have bread that rises is because we have baker's yeast, which is able to produce gases which cause the bread to rise. There's cheeses, you can have toxins, I don't know what you need them for, maybe for studying something in the lab, citric acid production, enzymes, and a lot of stuff. So what this slide is trying to show is that fungi are very important. There's also many plant pathogenic fungi. For example, you got Phytophthora infestans. It's technically not considered to be a true fungus. In order to be considered a true fungus, the organism must have chitin in its cell walls. So fungi have cell walls, similar to how bacteria and plant cells also have their own cell walls. It's just that the composition or what the cell walls are made out of are different. Chitin is specific to fungi, so that's how plants are able to tell if it's infected with fungi, if it can detect chitin. So you've got Phytophthora infestans, which is an oomycete, or an oomycete. You have here a potato that's infected with that. You can see it's pretty gross. There's also Puccinia, which causes rust on cereals. There is Magnoporthy, which causes all this kind of nastiness growing on rice plants. You've also got Eustilago, which infects corn. You get corn smell. You get this kind of disgusting looking growth on the corn. And some people eat it. Some people consider it a delicacy, but to each their own. And then you've got Colototricum, which causes anthracnose, which is similar in its symptoms to Magnoporthy. And then you got Aspergillus, which produces aflatoxins. Aflatoxins can be toxic to humans as well. So even after the crops have been harvested, you still have post-harvest pathogens which can cause the production of toxins on the food itself. So we have to be careful with some of these fungi as well. Alright, so some characteristics of fungi. They're eukaryotes, right? So they're actually more closely related to humans than they are to plants. And by eukaryotes, it means they have a nucleus and they have membrane-bound organelles. They're usually filamentous, meaning they grow kind of like I don't know, if you guys have seen the video game, the old video game of Snake, where you go eating something and it gets wider or longer and longer. It's similar to how it would grow, kind of serpentine-like, I would say. They can reproduce sexually and asexually. So usually the fungi that produce many types of spores are a result of asexual reproduction, just because you don't need fertilization in order for asexual reproduction to occur. The fungus can just keep creating more and more spores. And as I mentioned, they have a cell wall that's made of chitin and glucans, and they don't have cellulose. And the cellulose are the main component of plant cell walls. So again, they are cell walls, but they're just made of different things. They're heterotrophic. So what does hetero mean? The prefix hetero. Hetero means opposite. So opposite or different. So heterotrophic, trophy means energy, I believe. So it must obtain its carbon, it must obtain its energy from something else. So energy from something different. And they do not have chlorophyll, so they don't produce, that's the reason that they're heterotrophs. If they had their own chlorophyll, they could just use sunlight to produce their own nutrients. They'd be considered autotrophs, like plants. So let's see, the different types of fungi that we have. We've got biotrophs, which do not kill the host tissue. And instead, they penetrate the cell wall of the plant, but they don't penetrate the cytoplasm. So if you remember, there's a cell wall part, and then inside of that, before you get inside the cell, there's kind of an empty space. I think it's called the apoplastic space, but I could be wrong. Is it? Okay, good. And the fungus will produce what are called haustoria, which are kind of like a bunch of tree branches, which will sort of invaginate into the cytoplasmic membrane, but not directly penetrate it. And so the fungus is able to exchange all these molecules and nutrients from the plant so that it steals those nutrients from the plant cells. So those are called haustoria, or haustorium for singular. There's also necrotrophic fungi, which just produce a bunch of toxins and these enzymes that degrade the cell wall of the plant, and just make its contents leak everywhere, and then it just slurps up those contents. It just feeds on the dead tissue of plants. Then there's hemibiotrophic. What does hemi mean? Hemi means sometimes, or sort of. So sometimes biotrophic. A hemibiotroph starts off as a biotroph, and then later it's switched to a necrotroph. So it'll take as many nutrients as it can from the plant while the plant is growing, and then at some point it'll just straight up kill the plant once the fungus has grown enough. And then there's saprophytic, which just feed on dead tissue and don't kill the host. Saprophytes usually don't grow on living organisms, they usually just decompose matter. Alright, fungal structures. So we're going to look at the hyphae, which are the vegetative growth structures. And they kind of appear like, so you guys have been seeing how bacteria grow in circles. The hyphae tend to grow in that manner as well, kind of like just a big mass. And once you get a big mass of hyphae growing in a circle, we call that a mycelium. So yeah, that's that. Then you've also got septums or septa, which we'll see in just a bit. You got your hyphae, which grow in sort of like a long branch, and the septa will divide those branches into the individual cells. And then there are conidia, which I guess here they pronounce them or consider them to be the seeds of fungi. I don't know if that's very accurate. Again, I don't make these slides, I just have to present them. And there's many types of conidia or conidia-like structures, and they just produce spores. The spores are, like it says, the seeds of fungi. That's how you can think of them. And then there's fruiting bodies. The fruiting bodies are usually what you most commonly see. So something like this. I'm not sure what type of fungus this is, but this looks like it's a fruiting structure. You guys have seen it's been raining a lot lately, so I'm sure you guys have seen the fruiting structures of mushrooms. With mushrooms, if you look underneath them where they have all the gills, all the spores actually fall out from under there when a gust of wind comes. And it picks up those spores and spreads them everywhere. And that's why we have so many mushrooms on campus, because they're producing millions of spores. And over here on the next slide, we're going to see what the hypha looks like. So here we have a spore that's growing, and you're going to see the hypha start to grow out from it. You can kind of see the nuclei moving around. Some of these fungi have many different types, or many nuclei within one singular cell. And you can see another hypha starts to grow from that spore. And this video is provided by Dr. Brian Shaw, who works here in the plant pathology department as well. He does a lot of mycology related work. Here we have a mycelium. You can see you start off with one spore, and then it just starts to branch off everywhere until you get this large mat, and that's what we saw in the previous slide with these plates up here. That's the reason it looks like it grows in a circle. It's not really a circle so much as it's a bunch of branches radiating out from the center. And here on this infected tomato, you can see it's moldy. It's got all these fruiting structures coming out of it that'll release more spores to continue to infect other tomatoes or whatever's around that it can infect. So each hypha, or each type of hyphae, have different morphologies dependent on the classification of the fungus. So I don't think you guys will have to remember all these different types, but it's more of, for your information, so you can understand. Some hyphae, some fungi, they don't even produce hyphae. Like yeast, for example, they just start to bud off, similar to how bacteria grow. They just bud off and create a new one. Some have hyphal-like structures, but they're not considered to be true hyphae. That's why they're called pseudohyphae. And then you've also got cenocytic hyphae. Cenocytic just means it has no septa, so you can see there's no discrete cells. And then you've got septate hyphae, so you can actually see a septum in between that divides the different cells. And then you've also got certain types of fungi that produce septa with a clamp. And the clamp is just a way to, it's either for reproductive purposes or just to hold it together. I gotta admit, I'm not too well versed on mycology, but again, you guys don't need to learn every single type of hypha. Just so that you know, there are different types of hypha. So as we mentioned, there's aseptate, or also known as cenocytic, which is what the oomycetes and the zygomycetes have. You can have separate or without the clamp connections. Sorry, septate or without clamp connections, or you can have them. So those fungi that do have those clamp connections are usually the basidiomycetes. Septa. Now we're going to look at those structures that physically divide the cells. And there's different types of septa. They do have a pore in them, so they allow for the movement of nutrients and sometimes even the nuclei themselves. The nuclei can move between different cells. And let's see, they're used to separate older parts of the hyphae, and they're also used to classify fungi because the septa can have different morphologies. So this is just a close-up of what that mycelia looks like. Some are bunched together, some more mutualistic with plant roots. All right, you guys can read this on your own because it's pretty, basic stuff. And again, you don't have to memorize all this, but all that we're going to ask you to memorize are what the definitions are. So, for example, what a mycelia is, what a hypha is, what a septum is, rather than having to memorize the different types. It's just so you can see there's a lot of diversity. Even with the spore types, not all fungi produce a visible fruiting structure, visible to the naked eye. Like the fungus, the fungi that we see outside. But some of the conidia can have different structures, and you can see they start to all bud off. So, for example, the basidiospores that have from the basidiomycetes will produce spores in kind of like this fashion here from this basidium, which is different from a conidiospore over here. And the different phyla of fungi. Let's see. There's the Chytridiomycota, the Zygomycota, Glomeromycota, Ascomycota, and Basidiomycota. These last two right here, the Ascomycetes and the Basidiomycetes, are the ones that you most commonly see, the ones that are more recent, I guess you could say, taxonomically speaking. Alright, so let's finally get into today's lab. What we're going to do is we're going to infect some tomato plants with Agrobacterium, and I'll show you guys how to do that in the separate portion of the techniques video. We're also going to be looking at those potato slices that you guys re-inoculated for step 3 of Koch's postulates. We're going to see what you guys were able to get on your plates, and if you have any bacteria that are growing on those plates, at least different types, we're going to try to get a pure culture. Any questions then? All right, Max, let's do the techniques.